Hello students, today's topic is vegetative reproduction. First we have to know what is vegetative reproduction. It's a form of asexual reproduction occurring in plants in which a new plant grows from a fragment of the parent plant or a specialized reproductive structure. Why vegetative reproduction is known as a part of asexual reproduction? First, uniparental reproduction means from one parent body offsprings are generated. And next one is no gamete formation, which are the main functional procedure of asexual reproduction. For these two unique characters, first one is uniparental reproduction and another one is no gamete formation. That's why vegetative reproduction is also known as asexual reproduction. Then, different types of vegetative reproduction. Vegetative reproduction is dependent on two procedures. First one is natural and another one is artificial. Natural procedure is divided into four parts of the plant. First one is root, second one is stem, third one is leaves, and fourth one is other part of the plant. Whether artificial reproduction is dependent on some procedures: cutting, gutting, layering, grafting, and micropropagation, which is the main procedure of artificial reproduction or mostly used procedure of artificial reproduction. Now, stems are divided into three parts: underground, subaerial, and aerial. Underground stems are dependent on five procedures. First one is rhizome, next tuber, third one bulb, fourth corm, and fifth sucker. Subaerial, runner, offset, and stolon. And aerial is not divided into many parts. Its example is sugarcane and euphorbia. Now we discuss about natural vegetative reproduction briefly. We have to only recall the examples of different parts of natural vegetative reproduction. Procedure is not important because all the procedures are more or less same. First one is roots. Example: sweet potato, tapioca, yam, dahlia, etc. You have to remind these examples only. Next comes to stems. Rhizome, ginger, turmeric, tuber, potato, bulb, onion, garlic, etc. Corm, crocuses, gladioli, sucker, apple, banana. They are all underground stems. You have to remind it, they are all under the ground. Next comes to subaerial part of stems. First one is runner. Runner means basal internode goes horizontally and keep distance without mother's body. Example: Bermuda grass, peppermint. Next, stolon. Stolon is more or less same as runner, but runner goes horizontally and stolon grows obliquely upward. Its example is water hyacinth. Then we comes to stolon. Stolon means grows horizontally then emerges obliquely upward. Its example is strawberry and mint. They are all subaerial in nature. Then a special term is come, that is terror of Bengal. What is it? The plant water hyacinth. In Hindi, we called it jalkumbhi, and in Bengali, we called it kochuri pana. Its scientific name is Acornia species, and this plant is nicknamed as Terror of Bengal. Why it is called Terror of Bengal? Because it grows very fast and spreads on the surface of water body. When it spreads on the surface of water body, then it cut the sunlight and also increases the oxygen demand or biological oxygen demand or BOD, thus causing the death of fishes and other aquatic organisms. That's why water hyacinth is also called Terror of Bengal. Now, the leaf part of natural vegetative propagation. Kalanche, also termed as pathur kuchi, adianatum, etc. And others part of the plant body, utricularia, it's a plant. Agave potato or Dioscorea. This part means agave potato or Dioscorea is also known as bulbil, then lily. So, natural procedure is dependent on root, stem, leaves and other parts. Till now we discuss about natural vegetative propagation. Now we come to artificial vegetative reproduction. It's a complex procedure because it's a man-made procedure. It doesn't go naturally. First one is cutting. Cutting procedure is dependent on some steps. First, cut the stem just below the leaf. Next, dip the cut end into the rooting hormones. Hormones are IAA, mean indole acetic acid. IBA means indole butyric acid. Then the cut part is dipped into a container. After 4 to 8 weeks, the part of plant is well rooted. Let's see in the picture form. First, the stem part is cut apart. Then this part is dipped into hormones or rooting hormones. What are they? IAA means indole acetic acid, which is commonly known as auxin. Then indole butyric acid means IBA, which is artificial auxin. Then this cutting part is dipped into a container full of soil. After 4 to 8 weeks, the part of this plant is well rooted, means roots are emerged from here. This procedure is known as cutting. If roots are cut, then what is the example? Lemon, orange, tamarind. If stems are cut, then the example is China rose, sugarcane, etc. If leaves are cut, then begonia, kalanche. The procedure is same, only difference is the part of plants, root, stem and leaf. You must remind the examples of all the parts. Then come layering. Layering is also a artificial vegetative propagation or reproduction. It has 4 types. First one is ground or simple layering. Second one is tip layering. Third one is mound layering. And fourth one is serpentine layering. Now what is ground layering? First, bend a stem until its middle portion touches to the soil. Then this portion is pinned up with U-shaped pin with the soil. When roots are formed, then cut it from here and this is placed into another place. This is ground layering. Now tip layering. Tip layering means tip of the stem is dipped into soil, pin up here and cut it when the roots come out. This is tip layering. Mound layering means when many stems come out or emerge out from the plant body, then soil is covered up. When roots are emerged out from the stem, then cut the stems from here and then place it into soil. Serpentine layering means when main stem is emerged out, then it placed in the soil, pin up here. When roots are emerged out, then grows the plant. Then the plants again grow stems and then stems are again put into the soil. In this spherical structure or serpentine structure is known as serpentine layering. Main function is same, but due to different procedure, ground layering is typed into different types. First one is ground or simple layering, second tip, third one mound, and fourth one is serpentine. Now the example. For ground or simple layering, that is strawberry. For tip layering, blackberry and raspberry. For mound layering, that is mint. And third one is serpentine. What is clematis? You have to remind all the examples. Next one is guti. How does guti is formed? First, take a strong stem. Then epidermis layer means outer layer of the stem is removed from the stem. This part is applied with auxin or indole acetic acid. Then apply straw, soil, cow dung, and this portion is wrapped up with polythene bag. Spray water into this portion simultaneously. When root is emerged out, then the stem is cut from here and then the stem is inserted into the soil. Through this procedure, guti or artificial vegetative reproduction is performed. The example of guti is lemon, lychee, guava, etc. Now we focused on grafting procedure. Grafting procedure is divided into several procedures: wedge, crown, whip, approach, side. These 5 types is performed in any kind of trees. Procedures are more or less same except the cutting of plant. So you give any tree as an example. Then now we discuss the procedures of these 5 types of grafting. Firstly, we have to understand what is stalk and what is scion. Stalk means this part. This part which is attached with soil. This portion is known as stalk. And the part of other plant which is attached with stalk, that part is known as scion. This is a part of one tree and this is a part of another tree. This portion is cut from this tree. Then it is attached with the part of this tree. Cambium is attached and thus artificial vegetative propagation is performed. This is simply known as grafting. The part which is attached with soil, that is stalk, and the part of other plant which attached with stalk artificially, that is known as scion. Now we comes to different grafting procedure. First one is wedge grafting, where stalk and scion, stalk part and scion part cut into V-shaped. This is wedge grafting. Then comes to crown grafting, where stalk and scion cut into obliquely tongue-like structure, and then the cambium part is attached. Thus, the crown grafting is performed. Then another type of grafting, that is whip grafting. Here we cut the stalk and scion in exact tongue shape, not obliquely tongue shaped. It's exactly tongue shaped cut. So this is known as whip grafting or tongue grafting. Then comes to approach grafting. We found many colored flowers in same plant. This procedure is dependent on approach grafting. Two stems of different plants are joined together after removing their epidermis layer. Here epidermis layer of stems are removed. If the cambium layer of two plants are successfully joined, then it is known as approach grafting. Then side grafting. Side grafting is performed with the fat stalk and thin scion. Then they are taken and cut into V-shaped structure. Then the grafting takes place. So wedge grafting, crown grafting, tongue grafting, approach grafting and side grafting. These are the grafting procedures. Now we discuss about micropropagation period. First we have to know what is micropropagation. It is the process of multiplying plant stock material by growing plantlets in tissue culture to produce a large number of progeny plants and then planting them out. So now we have to understand the definition. First, we have to take a plant. It is a plant. Then we cut the plant part. This part is known as explant. So firstly, piece of plant tissue is cut. So firstly, piece of plant tissue is cut from the matured plant. That is known as explant. Then this cut part is cultured in a nutritious medium. Then Calais culture is started. Now the callus is formed. What is callus? Callus is an unorganized growing and dividing massing of cells in nutritious medium. This petri dish is full of nutritious material, means full of hormones, nutritious salts, and this is the primary cell which is divided into unorganized cells. Now these cells are put into another boyam. The boyam mouth is stuck with cotton to avoid air passing or any other germ passing. Then from the callus cell, each cell is divided through cytokinesis and development of embroid is occurred. From the each embroid, these are embroids. From the each embroid, single plantlets are developed. These plantlets are put into the soil and thus a mature tree is formed. This total procedure is known as micropropagation. First the explant, then the explant is put into a culture medium, callus is formed, callus formation occurred, then embroid is developed from the callus cells, then the embroid is turned into mature plant, which firstly known as plantlet and nextly it turned into matured plant. Now what is artificial seed? First artificial seed idea is given by T. Murashige in 1977. When we performed a micropropagation procedure, we found embryos. When embryo is developed, then it should be covered with gelatin or coraginan or caroginan. Thus, from the covered embryo, we get artificial seeds which are stored for another season or proper environmental. Now we comes to parthenogenesis and androgenesis. First, we have to understand what is parthenogenesis and what is androgenesis, means difference between them. Parthenogenesis means we got a ovum, but ovum turned into offspring without fertilization. This procedure is known as parthenogenesis, means ovum turned into offspring without fertilization. Example, Lacerta and Typhlina. Typhlina is a type of snake and Lacerta is a type of salamander. And what is androgenesis? Androgenesis is the same procedure except sperm is present instead of ovum. Here sperm without fertilization and offspring. That procedure is known as androgenesis. Now, parthenogenesis is divided into different types. First one is natural. Natural means without external condition, offsprings are produced from unfertilized ovum. In case of honeybee, is the best example of natural parthenogenesis. Honeybee, we got ovum. From ovum, we got drone. And drone are male honeybee. They produced by without fertilization. This is the best example of natural parthenogenesis. Now we comes to artificial parthenogenesis. What is it? With the help of external facts, means salts, urea are provided to them and offsprings are produced. Sea cucumber, it's a type of echinodermata or example of echinodermata. Parthenogenesis is performed by it. So, example of natural parthenogenesis is honeybee and example of artificial parthenogenesis is sea cucumber. Then haploid. Haploid parthenogenesis means offsprings are produced from haploid ovum, means N. Example, Spirogyra. Diploid parthenogenesis means offsprings are produced from diploid ovum. Example, Daphnia. So we all know that offsprings are produced by ovum in case of parthenogenesis. If offsprings are produced from sperm, then it is known as and the example of androgenesis is tobacco tree. So here is the complete explanation of vegetative propagation or vegetative reproduction. If you have any problem, then comment it below. I will solve it in next class or in the comment section. Thank you. See you in the next class.